Hi, my name is Ariana, and today I'm going to be talking to you about gene cloning as it relates to gene therapy. There are several diseases that have vanished or even decreased due to advancements in technology. For instance, smallpox and polio were eradicated due to the development of vaccines. Currently, there is a hope that in the future, diseases like diabetes, leukemia, and even Parkinson's, that they may be eliminated by this new technology or gene cloning. So you're probably asking yourself, what is gene cloning? This is when a specific gene of interest is located and isolated. Once a specific gene can be located in the original source of the DNA, there can be thousands of exact copies made. For the rest of the video, I will be focusing on one specific use of gene cloning, which is gene therapy. It is during this therapy that a normal copy of a gene is introduced into a cell in order to restore the function of a protein. One common element of gene cloning is a cloning vector. A cloning vector is a DNA molecule used as a mechanism to artificially carry foreign genetic material into another cell, where it can be replicated and/or expressed in that cell. There are 5 essential steps that are followed to create new DNA that will help with the process of gene therapy. The first step to gene therapy is to isolate the donor DNA fragment or gene. This can be accomplished by 2 different methods. 1, using a key restriction endonuclease enzyme, or 2, using a reverse transcriptase enzyme. Both of these methods create complementary DNA strands for the gene of interest. The second step to this process is the selection of a suitable cloning vector. The main role of a cloning vector is to replicate the inserted DNA fragment inside the host cell. The next step of this process is the incorporation of donor DNA fragments within the plasmid vector. During this process, a restriction enzyme is used to cut open the plasmid vector. At this time, the host DNA is inserted into the plasmid vector while the plasmid vector's DNA is taken out of the vector. The last step of the gene therapy process is the isolation of the recombinant cells. During this process, the host cells that contain the targeted gene are multiplied on a media. After this step is completed, the host cells are then able to be inserted into the individual or organism and able to be multiplied from there within the organism's body. This video was an overview of how gene cloning and therapy uses recombinant DNA to replace unhealthy genes with healthy ones. For the purpose of this video, I used viral vectors to give you a closer view on gene cloning.